blood parts. In order to give us a baseline for the discussion, I'm going to start by talking about the functions of blood. And then as we proceed through the conversation, I'm going to flash back to this list and I'm going to highlight for you what part does which function. And then if you understand the parts, what each of them do for the patients, it makes better sense to understand why we test blood the way we do, what the reasoning is behind the different counts we are meant to perform. Okay, so blood has the ability to carry towards all of the tissues everything that you see on the left. It carries nourishments in the way of minerals, in the way of electrolytes, of vitamins. It also carries hormones, which are messages that can produce the different organs to do different things. We carry antibodies, which helps us to control infection and disease. It also carries oxygen, which is critical for life. And it does carry specific immune cells, and those are the key to understanding if we will be able to address any one particular disease or another. And then through the act of moving through the vessels and the canals, it carries heat. On the right-hand side are two things that it takes away from blood, from the tissues, and it returns it back to the control center for the vascular system, which is a combination heart-lung combo. The lungs are there so that we can convert that carbon dioxide back into oxygen. And then the waste matter that it pulls from the different organ systems and tissues can help to be withdrawn as it gets to a different control center like the kidneys, right? So blood is a baseline for keeping everything status quo in essence, and it feeds all of the other areas of the body through a network of very, very large vessels called arteries, mid-sized vessels called veins, and then capillaries, which are very tiny vessels, but they infiltrate down into the deep, small narrows of tissue. And so when we're trying to look at blood, we could be looking at any or all of these things. And why we would look at them would depend on what are the concerns that the patient is presenting with. And then we know that we need to collect blood in order to assess the values of the parts, or a part in particular that's supposed to be doing one of these functions, in order to produce information for the veterinarian to use in a diagnostic manner. And that, for us as a technician, this slide becomes really important as the first step. We need to be able to read the list of requested tests, and then we need to be able to use our critical decision making to know which tubes we're going to collect for the tests. And if we don't know them off by heart, we're going to look at them on the lab requisition form, or we're going to review our notes, make sure that we know that if a particular test has been ordered, we know what tube we need to do it. Then we need to remember the order of draw. This is the second time you've seen this chart. It's a really great chart. It's an important chart. It's one that you should probably just have on hand until this becomes second nature for you to remember. And then we need to think about the volume of blood required to complete the tests. That requires our critical decision making, where we understand the size of the tubes, so we know what volume the tubes can collect, but also knowing if I'm going to be running a lot of tests and I need a lot of tubes, I need to mathematically collect exponential amounts of blood, right? If I have 1 3 ml tube versus 4 3 ml tubes, I'm collecting what could be 3 ml of blood or 12 ml of blood, and I would need to know that before I put a needle into a vein. And then we need to know how to handle the sample when we're done, especially when we know that some of the tests, or you're going to learn, some of the tests are happening in our hospital before we go to an external lab. Some of the tests are going right to an external lab, and some of the tests are never going to the external lab. And how we handle the samples and the tubes we use all will depend on those decisions too. So the most common baseline test that we have for blood to read, just a quick look at all of the functions of it, is to do what's called a complete blood count. And this slide tells you everything that's involved in a complete blood count, meaning we're looking at it in its entirety and as a sum of its parts. We want to know how many RBCs are there. We want to know what is the condensed volume of RBCs when we spin it down. And then we want to know what is the concentration of hemoglobin in the blood. And I'm going to get to why that's important when we talk specifically about that blood part in a moment. We also need to know how many white blood cells there are. And then we need to know how many are there in each of the different types of white blood cells, because the count is for all of them. The differential is there are 5 in total. How many of each of those 5 do I have? And then we also can count platelets. And in a CBC, especially a CBC using today's modern analyzers, a platelet count is typically just added in there because they're reading them off of whatever you've put into the machine. But we can also order additional specific platelet tests if we're really worried that clotting is an issue here. The collection, now that you know that the most common type that we need to look at everything is really a complete blood count, the things that you need to know is that we're probably grabbing at least 2 tubes. You're going to grab a tube of red. No matter what, you're going to grab a tube of red for everything, and red is the first one unless you're grabbing blue. So blue is a sodium citrate, and some of your analyzers will use a sodium citrate. It's more common for us to be using the red, the purple, and the green. So if you're pulling a CBC, you're going to pull blood enough to do a red tube, and then that'll help you separate the blood out into its parts. The second thing that you want is a tube with an anticoagulant in it. EDTA, the lavender top tube, is the best preservative of cell quality. And so grabbing a purple top tube is super common for any clinic, because we know that once we make a smear and we want to look at the cells, anything having been preserved in EDTA, we're pretty assured that those cells have been frozen in time because of that anticoagulant. In order to make sure that we have the right concentration of anticoagulant to blood, we never want to force blood into a vacuum tube beyond its vacuum. We also don't want to withdraw less blood than what is required, because otherwise you'll have an improper ratio or an improper mix. And if the dilution is wrong, then the EDTA can actually can be inhibited from doing its job. If we don't give it enough blood to work with, it can actually be too much anticoagulant, and it can just change the way the cells are going to look for us. Once we've got the blood in the tube, the handling that we need to remember is we have to have a soft or gentle mixing of the anticoagulant with the blood. We're not shaking this thing like a salt shaker. You can roll it between your 2 palms, but you really also want to roll it and then tip it so that it's rocking, so that you've got a nice mix or a nice coat of blood on the inside of the test tube, and it's collecting all of the EDTA that was also loose in the test tube, and it might have been in droplets on the side of the tube somewhere up near the stopper. But by gently rocking and inverting the tube, you can coat it, and you're getting a nice good mix. You also want to check your purple top tube to see that no clots have accidentally been formed. 2 things can happen when you have a clot. The first is that the clot can mean that there's something wrong with the collection, and then all of the cells will be, it's a cue that all the cells could be compromised if the platelets are compromised and the cells are compromised. The second is that if you try to run this kind of blood in an analyzer and you happen to put a clot in the analyzer, you could be wrecking your analyzer. Reasons why these kinds of things can happen could be that we had a difficult time getting the blood. The animal was fractious. They were afraid, they were scared, they were panicked. We tried to do low stress handling. It took a long time. And because of that, we know we might have some changes to the way the blood will work. If the animal moves when we're in the vessel, which is where, you know, knowing your fear-free and low stress handling is important. If we're following those principles, and if we absolutely had to use restraint for the safety of the animal, and we did it properly, we can avoid things like platelet clumping. We can avoid things like shredding of the vessels as it passes through the tiny little hole of the end of the needle. They can tear. They can also tear on their way from the needle into the vacutainer because of the power of the suction. We also could run into issues with not being able to get enough blood, especially if that's a difficult venipuncture, or when we're putting it into the tube, we don't mix it properly. We put it in the tube, we rock it back and forth twice, we lay it on the counter. You haven't necessarily done what you needed to do to make sure it's a good mix. And so clots can develop in the areas of the blood that were not touched by anticoagulant yet. And then even if you pick it up in, 5 seconds later, you've already given that blood 5 seconds to try to clot itself, and it will. And then when you try to mix it, the anticoagulant can't break down a clot once it's formed. It's just going to have to stay there. Other issues that we could create as a technician, which is the reason why I'm highlighting these are things we can control. So the good news is that these are things that can go wrong, but knowing they can go wrong, we can put in measures to make sure it goes right. This one has got to do with handling of the sample. So if we have successfully taken the blood from the patient and we're not handling the sample well, we could accidentally mix it with water. This is specific to, say, making a stain or a smear. If we've put the blood on a slide and then it accidentally gets a droplet on it, you're going to know the difference when you see the look of the red cells specifically. They're going to look all rotten or diseased. And it's not that they are diseased, it's that you've changed what happened because they were touching water. And the osmotic pressure and the osmotic differences between an RBC and water caused it to look toxic. The same thing can happen with rough handling, just like in bad venipuncture or a bad collection. We can actually handle it inappropriately in such a way that we shred the cells ourselves. So the third thing that could go wrong is that we don't store it properly when we're done. If you have put the tube, you've collected the blood well, you've handled it properly, it's in a vacuumed tube to the right level, it's mixed well, eventually samples will start to age naturally. If you are going to use this sample, you need to use it within an hour, fresh from body at room temp, or you need to put it in the refrigerator. And then you can leave a sample in the refrigerator for up to 7 days. But after 7 days, trying to read a sample after 7 days, even if it's an anticoagulant and you did nothing wrong, we're going to still see some cell death. And this is a picture of what cell death can look like. So we won't hang on to blood for long periods of time, which makes the first hour really important for handling and collection, because we can create, say, a smear out of the blood to preserve what it looked like in that moment. And once it's been permanently preserved on a glass smear, those can last for decades. And I actually have smears in our hospital that you're going to be looking at that are decades old. I think there might even be a smear that's as old as I am. We can also see issues with the blood starting to crystallize, or properties of the blood crystallizing, and the platelets start to clump and all the cells look like they're dying. So this is what old blood looks like. Here's a picture of that, of the other issues that happen with storage. So if we've created a smear, we decide we're going to stain the smear so that we can better see the differences in the cells. If the stain we've used is dirty, it leaves little globs all over the slide. Just comparing the slide on the left to the right, you can see that there's kind of little flocculence, like little dusting of purpleness all over. There's really dark globs of purple, but then there's also these little light globs of purple all through the background. And you'll notice it's missing on the right. It's because everything you see on the left is dirt from within the stain that got attached to the blood sample on the smear as you dipped it in. Whereas water, remember I told you water makes the red cells look dangerously infectious? Look at that. Look at those red cells. I know you haven't learned what normal red cells look like yet, you're getting there, but the hole punched effect, or the fact that they look swollen and rough, just take a look at the red cells on the left. Those actually look like pretty healthy red cells, and the ones on the right don't. Oh, I see the question in the chat. Thanks, Mandy, for answering. Yeah, 4 degrees is usually a good temperature for your refrigerator. Anything below 5 degrees means that you're cooling without freezing. We don't want to freeze blood. If you get closer to the 0 mark, you have the potential of freezing blood, and that definitely will destroy it. It has to be chilled but not frozen, unless it's a fast freeze, which is why blood products from, say, a blood donor clinic, like the Canine Blood Donor Clinic, they take it and process it a different way. So one of the things we do to get around all of those issues is we collect the tubes, we collect them in the right order, we collect them with the right volume, and then we immediately take a sample out of the purple top tube, which was mixed with the anticoagulant, and we make a blood smear. We put a drop of blood on a glass slide, we drag the blood across the slide so it's really, really thin, and the cells have space between them, and then we dry it to the glass and we put it through a 3-step stain system. And you're going to do more of the 3-step stain system next week. This week, you're learning about making a smear in general. And it sounds easy, and even the videos make it look easy, but it is not an easy thing to do. So cut yourself some slack. You're teaching your body to do something new. And once you start to get a consistent formation of smear patterns, it's just a repeat and a repeat and a repeat. And once your body gets used to it, you will catch on, but it will take you some time of repeating blood smears before you feel really good about it. So don't beat yourselves up if in like 2 minutes in lab you can't get a good smear. That's okay. We're going to get you there. Once we get these smears down, it does help us to keep them permanently so we can see what the blood morphology looks like, all the different parts. They've frozen in time. Part of the staining includes a fixative that keeps that blood attached to the glass before we add the dyes. And then we can put a cover slip on it and it's there forever. You'd have to take the slip off and scrape off the blood to try to destroy it. So you really do have a permanent snapshot. And that way, even if you had to put the blood tube in the fridge and 7 days go by, you can't run the tube blood anymore, but you can always look back at the smear if you need to. So when you're looking at the morphology or the shape and the size, you can then decide, are these blood cells normal? Are they abnormal? You can use that information to confirm if you used a blood machine and it gave you funny results. You could look at the smear and say, oh, I see now why the funny results are there. So it gives you a bit of opportunity to quality control any of the automatic analyzers that you're working with. It also gives you an opportunity to look for things like parasites, which none of these analyzers are good at. They're really only good at looking at the blood cells. And even then, it's a computer. So the way that your analyzers look at the quality of a blood cell is like Blackboard grading your math tests. Sometimes it's going to get it right, sometimes it's going to get it wrong, but it always helps if somebody runs their eyeballs over it. So just like you want me to run my eyeballs over your tests in math, you should always be double checking your smears. Even if you're using a really high quality, expensive analyzer, look at a blood smear. Nothing will compete for your critical analysis of what normal healthy blood looks like to abnormal not healthy blood. This is how you make a blood smear. This is why it looks so simple, is that I've got 3 steps and a pretty picture, and it kind of seems like it should be easy to do. I'll tell you the stages. You're going to drop the blood on the glass, and you're going to drag another glass slide, a secondary glass slide, back into the blood. It's going to be held at like a 30 to 45 degree angle. And then that's usually the first step students get worried about, is they don't wait long enough for the blood to stretch across the spreader slide. It's like all of a sudden your heart starts to beat like you're being timed. I'll tell you what's going to happen is that even if it gets to the edges of the slide, nothing, you're fine. So don't panic, just watch the blood go. And as soon as it's stretched, then you have to do it in one fluid motion, but it's not a race. You're not racing to the end of the slide. You can have a controlled push with your spreader slide, control the push forward and straight. And if it's a nice forward straight control, you're going to hit the end of the slide in less than a second. But what's going to happen is the contact point between the two glass slides becomes like this tension with a meniscus with the blood. And as you're pulling the spreader slide from the original drop, all the blood is getting thinner and thinner and thinner. And then eventually there's no more blood to stretch and your spreader slide disconnects from the blood completely. And you're left with this really beautiful, curved tip. And looking down at the slide where this curved tip is and this feathered edges, you will have a great counting area where the cells are not so close together that they're on top of each other. And that if there's space between the cells, you can then really look at the shape of a cell and the size of a cell because it's not being affected by bumping into its neighbor. Okay. So after you've made a slide, you want to dry it pretty quickly. In the video that I shared with you, they've got a hair dryer. We also have a hair dryer in the lab. It's used for this and other things, but you can also just lift up the slide and you can wave it in the air back and forth. This is one time where I do want you to shake blood, but it's already been stretched on a glass slide. So you can move it back and forth. Don't blow on it, just move it back and forth so that you're drying the blood to the glass. And then from there, you have 2 days from the moment you made the smear to stain it. If you wait longer than that, then you haven't fixed the cells in their state and you could collect dust on top of it or they could change. So within 24 hours, you're going to dip them in stains. We're going to talk about that a little bit next week. And then you can either leave the slide on the counter at room temperature or put it in a slide box. Or if you do have to send the slide to an external lab, you can prepare it for shipping. They have these little miniature foam boxes, looks like tiny little treasure boxes with little slots inside them, and you can slide your slides in there. They come in hard plastic too. They also come in cardboard, and any lab that's asking for a smear will give you their little transport box that they want you to use for free. And then you put your slide in it and off you go. Just a note here that if you are shipping blood smears, formalin, which is a tissue preservative, will impact blood even if you've smeared it and even if you've stained it. Formalin gases are really, really strong. Formalin in general is really strong. It can be considered cytotoxic if the tissues are living. So what you want to do is, if you're sending away a tumor and then you're also sending away a blood smear, if your tumor is in a container of formalin, that is a package all by itself. And then you create a separate package for any blood and blood products to go to the lab. So here again is that picture of where we would look to count our slides. And when we're looking at our blood on the slide, we'll be able to discern that there are 4 different parts to blood that we are caring about. Remembering from the original list of the things we test for a CBC, we are testing for item number 1. What was the first thing on the list that we tested for in a CBC? Item number 1, take a look at the picture. Yeah, red blood cells. Oh, thanks, Amelia, you're right on it. The first one's an RBC. Yes, it's a red blood cell. The second one is a WBC. Those are our white blood cells. The third one is plasma. Plasma is the background of the blood. It's the liquid, it's the juice that moves through the vessels, and it's a conduit for everything else, because the red blood cells and the white blood cells don't move by themselves. They have to be suspended in plasma to get around. Same with the platelets, which is item number 4. Those things are suspended in the plasma and the plasma becomes the vehicle. So we're going to talk about each one of these parts individually now, and we're going to talk about the tests we run to assess their value. So when we're talking about an RBC, here's a picture of a screen of just red blood cells. They are also called erythrocytes. I know you're taking etymology right now. Ereth with the R is red. ERYTHR always means red. Okay. O is the combining vowel. Site is a root. And so we're talking about red cells. It's right there in the name. This is what red cells do. They carry oxygen towards tissues and they carry carbon dioxide back. They do that because they have hemoglobin, okay? And so part of what we're looking for when we're talking about red cells first is to estimate the percentage of red cells that are found in the blood. And the fastest test to do that is using those microhematocrit tubes that you guys were practicing with. Because as soon as you draw just a little bit of blood up in one of those tubes and then you push it down in a little plasticine, you can put it in that centrifuge, balance the centrifuge and spin it. And it doesn't take very long to spin it. A PCV spin can be 2 to 3 minutes, depending on what kind of centrifuge you're using. Really short period of time, sometimes 30 seconds, depending on, again, what kind of tube or centrifuge you're using. And as soon as you're done that, it'll split. The bottom part will look red because that's where all of the red cells went. They're down near the plasticine end and they're heavy and there's a large volume of them. In fact, it's probably about 40 to 45% of the tube itself is red cells, or it should be in a healthy animal. And that gives you just a hint at how much hemoglobin should be in the body if you've got the right number of red cells. Now, if we're dealing with a patient that has anemia, anemia being too little red cells, what do you think is going to happen to that packed cell volume? If a regular packed cell volume shows about 45 to 50% of your PCV is red cells of your tube, yes, it's smaller. So if you've got a patient with low red cells but lots of plasma, we should be suspicious that there's not enough red cells in the patient in general, because the plasma and the PC and the packed cell volume of RBCs, they should be about the same. Right in the middle is where the white cells are. And there are white cells in the body, quite a fair number of them, but clearly not as many as there are reds. And the white cells will rest on top of the reds, and it almost looks exactly white or creamy colored. It almost looks like a little bit of plasticine ended up in the middle of the tube, but it didn't. Those are actually white cells. And then at the very top is plasma, and plasma contains everything that's been dissolved. It doesn't actually have a body shape to it, but it is a part of blood. And so when we're looking at our tube, when we're done with it, by lining it up on our card reader, we get a pretty good idea already of what the average percentages of RBCs are in the body. And that whole test can take you less than a minute. Well, no, that's not true. Less than 5, depending on your centrifuge, right? If you got an older model centrifuge that's spinning at a slower RPM, you might need to spin your PCV for up to 3 minutes. But if you got a new centrifuge that's spinning at a higher RPM, meaning faster rotations per minute, you could finish this test quicker. So I love that this lined up, and it's only because I teach both of your classes that it lined up. But this week, when we were talking about scientific notation last week and you were seeing things expressed, this week is now giving you the truest reason as to why. Look at how we express red blood cells. We can express them as liters, or we can express them as microliters. Even looking at the exponents, a liter is times 10 to the 12th, and a microliter is times 10 to the 6th. You can tell that there's 6 decimal places or 6 places between microliter, which is that little U, and a liter. So a liter is 1,000,000 microliters. And depending on the machine you're using, the province or state you're working in, and the patient, you're going to be expressing your counts as an RBC times 10 to the 12th per liter or times 10 to the 6th per microliter. 10 to the 12th is pretty common around here. It's really hard to count red blood cells manually. In fact, we don't do it anymore. We used to. But we used to when I graduated, I had to learn that way. But nowadays, technology has become so efficient that we don't make it incumbent on the technician. Your time is better spent doing other things. So you will get an actual RBC count from your electric or automated analyzer, and instead you're going to be able to estimate it from your PCV tube. And that should give us an idea about the hemoglobin level, because hemoglobin sits inside the red blood cells. But that's not to say that it's accurate. We're just guessing, right? We're assuming that each of the blood cells that you are noticing has the same amount of hemoglobin, and the hemoglobin is accurate for the size of the cell. So we can estimate in grams per deciliter of blood how much hemoglobin there is, but the better 3 tests are run using our automated analyzers, and we need 3 pieces of information, 2 specifically, but 3 helps. The first is we need to figure out how big are the RBCs. Your automated analyzer will do that. It's called mean corpuscular volume. So we're trying to figure out what the average volume is of each cell, each red cell. Then we're trying to figure out what is the average volume of hemoglobin per RBC. And then we can figure out how much hemoglobin does the patient have, given the number of cells they have, knowing how big they are. All of this is done automated. We're not counting these by hand, but you can see how it would be important. If we have a patient that comes in and you're questioning if their body is getting enough oxygen, and you're running a packed cell volume and it says they have enough red cells, the question is, why do we have enough red cells, but they're not actually carrying enough hemoglobin? And the answer is there are poisons that do that to animals. There's disease processes that do that to animals. So just knowing how many reds doesn't always tell us how much hemoglobin is there. And if there's not enough hemoglobin, you're still not getting oxygen moving through the body, because that's the carrier. Do I have any questions before I move into other cell parts? Or do we feel good about reds? Feeling okay? Okay, so in essence, when it comes to reds, we're going to run the PCV and it's going to give us an estimate. We also know we've created a blood smear, so we're going to be able to look at the health of them, and that's about as far as it goes for us as a technician on our own personal counters. The other tests are we're using automated machines, and it will depend on our sample collection and handling to make sure that these are accurately represented. So let's now think about the other parts of the blood. The second part we had was WBCs. So if erythra is red, and cyte meant cell, and O was a combining vowel, what do you think leuc means? Yep, leuc means white 100% of the time. So you got leukocytes, and then you got erythrocytes. This picture has still RBCs. I can't, and hopefully you don't ever come across a patient that has only whites and no reds, because that would be terrible for them. But this image has actually all the parts. The background that looks like faint purpley is the plasma that's picking up stain. The red or pink shaped circles, those are the red cells. The white cells are the purpley shaped ones with a darker purple nucleus. And then you see also little purple specks on the background. Those are actually your platelets. So let's take a look at what kind of things white cells do. This is, in fact, the entire category. Every one of the white cells is an immune cell. They have a specific job. They live for a specific period of time. We are going to talk about each white cell one at a time after the study break, and you're going to get really good at understanding what a normal white cell looks like for each of those normal white cells. And they come out specifically to work on infection. So they're constantly circulating in the blood, ready to address infection at any given time. And as we're reading the blood smear and counting the white cells and looking at the health, we're going to get a good idea of the body's ability to fight infection. We can also see if we've got a high number of one kind of cell, maybe there already is that kind of infection somewhere, which is why that cell has a higher number in the blood. The body put more of them into the blood because there's dealing with an infection. And depending on which cell we see more of and the health of that particular cell, we can actually tell an awful lot about what is happening in the body. So we will want to know how many WBCs exist in the blood. And that is a count that we will do ourselves using a smear. We also want to get us an idea of the estimated blood volume in the entire body. And using a PCV tube helps us because remember, we had the plasma on top, we had the buffy coat in the middle, which is the white cells, and then we had the red cells down at the bottom. We can use the same scale, the same card to read the buffy coat and to read the plasma. Now, based on today's discussion in math about ratios, I could create a statement that says, for this volume of plasma, I had this volume of reds and this volume of whites. And that would be a great way of comparing parts of a whole. We can definitely use an automated analyzer, and they're getting really good in their technology. There's a couple different kinds of analyzers there. We're going to learn more about them specifically next week that we're going to use, and you will learn on the analyzers that we have on campus specifically. And we also, through the advent of science, are getting better able at reading different species types using those analyzers. There was a time when, you know, you could only get blood normal parameters for cats and dogs, and now then they expanded, and now there's horses and there's sheep. Even they've got sheep or cow normals, and you can even get them with exotic animal normal value sets, so that when you're running the blood, the computer can flag whether something is elevated or underrepresented based on the normal set of the species you choose. So in order to count the cells, we're going to work on a ratio system. We're going to work on counting 100 white cells, and then we're going to use math to express that if in 100 cells, I find, looking in these classifications, I find X number neutrophils, X number eosinophils, X number lymphocytes, X number monocytes, and X number of basophils, I'm going to know that if I was to take 100 leukocytes and I wanted to report it in a larger number, I keep the ratio. And that's how we're going to deal with white cells, because clearly we're not going to count through a liter of blood like we represented it for RBCs. That doesn't work. We're not going to pull a liter of blood on our patient. So what we're doing is taking a small amount of blood, using it as a baseline, and imagining that this little slice of blood, I could take a liter, make it into equal slices. They would all relatively have the same amount of cells every time. This is just an image, a drawn image of what each of these cells look like. As we go through the semester, we're going to spend time talking about each one of these cells specifically, what they do specifically, what they look like when they're normal, their sizes, their shape, and then you're going to practice finding them using your microscope, which is why I spent a lot of time making sure you knew how to use your microscope, because now you actually need to rely on it. And as we count using that differential counter, we'll be able to count both at the same time. We don't just sit and count 100 blood cells and then go back and count the neutrophils, the eosinophils, the lymphocytes, the monocytes, and basophils. Instead, what we do is we sit at the microscope, we use the differential counter, and every time we see a neutrophil, we push the button, and the total count rises until we've got a total 100 based on all of the rising counts for each category. Any questions about the white cells before I move on to plasma? We have two more parts to talk about. We have plasma and platelets. I just want to pause in case, no, still okay? Okay. So plasma, this is human plasma that you're looking at. Animal plasma looks the same. It might be just a little bit less yellow in my personal opinion, but I think color can be slightly subjective to our eyeballs. There is no other way to better describe it than to show you what it does. It is the liquid that holds the dissolved pieces and moves it through the body. It is the one thing that we're not spending our time focusing on under a microscope, yet it does so much. It carries all the minerals, it carries the electrolytes, it carries the hormones, it carries the vitamins, it carries the antibodies, it carries the waste matter. And by virtue of moving through the bloodstream, it is what conducts heat inside the patient, right? So if you have, if you've been out on recess, thinking back to the days of when you used to have a recess and you went to the recess teacher and said, I'm cold. And she said, do some jumping jacks. Or maybe those are just my recess teachers. That's why. If you get your blood moving faster, you're going to start heating up from the inside. And it's because the plasma is moving around. And it just happens to be carrying the other things like the cells, but it's really the function of the plasma that keeps them warming. Okay. So, super critical part of what the patient health would be to understand what's happening. And for all of these parts, especially, well, not so much the gases, but those are attached to cells. But for all of these parts, like the vitamins, the minerals in the nourishment part, the electrolytes, they all contribute protein. They're dissolved, you can't see them, but they're proteinaceous inside the plasma. And what that means is protein is thicker than liquid, even though you can't see it. And so while it's in the plasma, it changes the way light moves through it. And the best way to see that is through your refractometer. So again, that one little PCV tube does so many good things for us, right? It gives us an estimate of red blood cells. We can start to predict hemoglobin, gives us an estimate of whites, and it tells us roughly how much plasma we've got. And then we're still not done with it. We just snap it off and we dump a little bit of that plasma on our refractometer. And then we can get an idea of how much protein is in it. And that gives us an idea of, are there enough minerals dissolved in the plasma? Are there enough electrolytes in there? And if the protein is accurate to what we expect for a healthy animal, we'll know or presume, yes, it is. And then we can take an analyzer to go look at each of those different areas specifically. But for now, with that 1 5-minute test, you've said so much information. So in addition to using a refractometer, we would also look at the color of the blood. We would the plasma. We would describe it. Is it yellow like this? Is it straw colored, like a little less yellow? Is it clear or is it cloudy? Is it white? Is it green? Those kinds of ideas of words to describe the color or the clarity of the liquid help a lot as well. You'd have to imagine the patient would be pretty sick to see plasma that has pink in it. But if plasma has pink in it, that tells me that red blood cells are shredding somewhere, and it's shredding inside the animal if the venipuncture was right. If we did a nice clean poke, it went into the tubes fine, the patient looks fine, and the plasma is pink, it tells me it's picking up pieces of red cells. They've been shredded and they're dissolving into the plasma. Why though? That would be where the disease process comes in, right? So we want to talk about that. And then when we put our samples into our automated analyzers, they can do more to tell us about the actual breakdown of components inside plasma, including if there's clotting proteins in there as well. So the last thing that we want to talk about as a part of blood that we can't ignore and we will be analyzing are platelets. Another word for platelets are thrombocytes. The image that you see does have a white cell right in the middle of it. It's actually a neutrophil. So you can get used to seeing those. And then the staining is a little bit different on this sample. You can see it's a little more blue tinged, but all of those purpley colored spheres that are just a little bit smaller than the white cell and don't have a nucleus, those are reds. And then in between that, you see, it looks like little pink ghosties. They're circles and they sometimes have little bluish arms coming off of them. Those are all platelets and they're all coming towards that white cell. And it looks like they're going to start to form a clot around it. See them all coming in together? As they're loose in the body, they're actually small little round, what looks like a plate, small little round cells. But as they become active and they're working, they put out these little projections, these wispy little projections to hang on to each other and hang on to everything else, because they are not performing any one of those functions. Instead, they are a net, which is why I put that net there. The only job of platelets is to keep the blood inside the body so that all of those functions can happen. They are a mechanical feature of blood, but really important. Because if the body is leaking blood, it is no longer able to carry nourishment, electrolytes, hormones, vitamins, antibodies, heat, and oxygen with immune cells over to the tissues. It is also incapable of collecting waste matter and carbon dioxide. So the blood can't be leaking. And the platelets are that net for us. And so when we're talking about the number of platelets, you can estimate based on what you're seeing when you're looking at a smear, because you'll see things like this. You'll be able to take a look and say, wow, I see the platelets coming together. They look like they're going to form a clot. I don't really see a clot yet. There's maybe 4 that are close by in the 2 o'clock position. There's 4 that are kind of glomming together, starting to create that net. But for the most part, they're kind of loose in the body. But if you really want to know if your platelets are working well, and if your body is clotting well, you can use an analyzer to run that value, or I wish I had the capacity just to jump to one particular slide. Close your eyes if you get sick really quick. I'm just going to go backwards really fast. Well, before I do that, look at this picture. It's a great picture. It shows you those spinous projections. There's little fingery projections that come out of platelets when they're activated. All of the little white balls, those are platelets on this picture. Everything, and then the reds are looking like reds from inside the picture. Okay, now I want to go all the way back to here. I'm now making myself dizzy. Okay, this table, the very last one, the gray tube, potassium oxalate or sodium fluoride, those are for coagulation testing. The potassium oxalate is a gray top tube specifically for clotting factors. So if you have a patient that comes in and you think they're von Willebrand's, like a Doberman, a von Willebrand's dog is like a, they have, I can't remember what, oh my gosh, isn't that sad? I cannot remember what the disease is in humans where they can't clot blood. Please help me. There's a disease that humans, more common in males, hemophilia. Thank you, Shelby. So von Willebrand's and it's more common in breeds like Dobermans, is a clotting issue similar to hemophilia in humans. And you can have gray top tubes in your practice where you put your blood in them and then you continue to invert as you time it, and you time it to the second how long it takes for a clot to form. And you can use glucose testing for sodium fluoride. So if you know you're going to want to do a von Willebrand study, you can pull a gray top tube after you've done your red, after you've done your lavender or your green, depending on what analyzer you're using. They may want you to use that heparin tube instead. And then you can run your clotting factors. And through that, we're going to be able to really assess all of the blood functions real quick, just pulling some blood, dropping it in a few tops, and then making a smear.